Dear students, I am Dr. Amit Gangwal, Associate Professor at Shri Vile Parle Kelauni Mandal's Institute of Pharmacy Dhule. In this video, we will talk about herbal dosage forms, that is traditional or classical Ayurvedic dosage forms, and we will see the basics of NDDS, that is novel drug delivery system in general. And we will also see the importance and need of NDDS in general. Then gradually we will shift to herbal NDDS. In that we will see only phytosome as per the syllabus given in herbal drug technology by PCI for B Pharm third year, that is B Pharm 6th semester. So majority of the content has been taken from this book Herbal Drug Technology by Nirali Prakashan, and this video is not for commercial purpose. It will be uploaded on the YouTube for benefits of the students and other public who is interested in this particular topic. And few content, I have some content I have taken from this review article, that is Phytosomes as innovative delivery systems for phytochemicals, a comprehensive review of literature. Now, so in corona we have seen doctors have recommended paracetamol 650 milligram tablet, and the market leader in paracetamol is Crocin, and other brands are also there, like Calpol is there by GSK, Crocin is there by GSK, then other brand is Dolo by Microlab, Dolo 650. So it got very high momentum. It became talk of the town in corona time, and even after corona time. That is Dolo 650 milligram tablet. It contains paracetamol 650 milligram. So as per the information available on various websites which covers brands and branding strategies, according to them, this quarter page advertisement in Times of India and even in Bhaskar, I think, in this commercial, the company is saying that Crocin 650 is the trusted paracetamol brand that starts working in 5 minutes. So they have given the logic why it takes 5 minutes, why it is so fast in action. They have mentioned in this subtitle, you can see here, this one. If you think all paracetamol tablets are same, think again. Crocin 650 milligram with scientifically proven Optizorb technology is an advanced formulation that dissolves in your stomach faster than standard paracetamol tablet. It starts working in 5 minutes. So they are trying to be superior. They want to place the product ahead of other 650 milligram brands. So they are using the scientific technology, that is Optizorb. So according to a market a firm which studies all type of advertisements, they are saying that it is unclear who is Mr. Khanna in this ad. You might be seeing that in this newspaper advertisement, the person is Mr. Sachin Khurana. So you see, they are telling that we don't know who is Mr. Khurana. If he is doctor, then it should be doctor should be here in prefix. If he is not doctor, then why apron is there anyway? So because of through this aid, they are taking a jab at Microlab's popular paracetamol brand Dolo 650. This Dolo 650 has witnessed an unprecedented, like never before, level of popularity during corona time. So through this advertisement, I have tried to show you how technology takes the company or brand ahead of the rivals or the competitors. So types of herbal dosage forms, basically we can classify in three ways. Traditional dosage form, that is Asav, Arista, Bhasma, Churna. Modern herbal dosage forms like tablets, syrups, etc. Novel dosage form like phytosomes, liposomes and others. So we all know Asav, Arista are hydroalcoholic or self generated alcoholic preparations in which alcohol is generated through the process of fermentation. And bhasma are nanoparticle type of age-old Indian Ayurvedic formulations. Churna simply type of mixtures. So we are moving ahead now. We will compare classical or conventional herbal dosage forms and novel dosage forms, right? So in case of conventional herbal dosage forms, drug is rapidly and completely released after administration. In case of NDDS, drug release can be modified for extended period. So this is the basic difference. Now we will talk about the second difference, that is absorption is rate limiting step. In case of NDDS, rate limiting step is a drug release. You can manage or control the type and the rate of drug release. So in case of conventional formulations, there is blood level fluctuations is there. In case of NDDS, constant blood level can be achieved. Similarly, in case of conventional drugs, patient's compliance may not be there. In case of NDDS, more patient compliance is there. These are few differences or points to compare classical herbal dosage forms or even classical dosage forms with NDDS. So what is the meaning of herbal formulation? Herbal formulations are the dosage forms which consist of one or more herbs or processed herbs. We all know usually herbal formulations are polyherbal in nature, means they contain more than one plant material. So they are called polyherbal, right? So they are processed herbs in specified quantities to provide specific nutritional cosmetic benefits meant for use to diagnose, treat, mitigate diseases of human beings or animals. They can alter the structure or physiology of human beings or animals. Now, NDDS means formulations, systems, or more importantly, technologies for transporting a pharmaceutical compound in the body as it is needed to safely achieve its desired therapeutic effects. Why we are using the word technology here in the definition? I will explain you through some example in next few minutes. So drug delivery systems are based on approaches that are interdisciplinary and that combine pharmaceutics, bioconjugate chemistry and molecular biology. Obviously, to design NDDS, you should have in your team, you should have material expert, you should have ecology expert, you should have definitely the pharmaceuticists, that is the experts of formulation, and of course if it is herbal drug, there can be need of natural chemist or pharmacognosist. So it is a novel approach to drug delivery that addresses the limitations of traditional drug delivery system. So what is the most common limitation of traditional drug delivery system? You cannot control the rate of release. Once you have taken, for example, a paracetamol tablet of 500 milligram, then once it goes in the body, it will start releasing the medicament. But in case of NDDS, you can have the control on rate of release, site of release, and onset of release. When to release the drug, it can be in your control. How to release the drug, it can be in your control. Where to release and how long to release, they can also be in your control. This is the beauty of NDDS. Now, as I told you, we will talk about why there is need of novel drug delivery system, that is NDDS. So you see the portions I have highlighted in yellow color, that is need of NDDS. The first, to get desired release profile, that is controlled release or sustained release, means after taking the medicament, how many milligrams of the drug should be released in first 20 minutes or first 1 hour, or for example after 2 hours, how many milligram of the drug should be released. That is the desired release profile. Then site specific drug delivery. For example, in case of cancer treatment, you need to release the drug only at the site of tumor, and your non-tumorous cell, that is the healthy cell, should not be affected by the chemotherapy. That is to deliver the drug at the site of action. This is not possible through classical or conventional or traditional type of drug delivery system. And third point is to lower the drug. If we are devising some system in which drug will directly go to the site of action, so during entire journey the metabolism or the wastage will be minimum. So obviously if minimum journey is there, minimum metabolism is there, then of course you would need low amount of the dose and reduced or minimum possible side effect. And fifth reason is increased bioavailability. Now, with the advancement in different scientific techniques of preparing formulations, novel dosage forms are being developed to overcome the limitations of conventional dosage forms such as tablets, syrups, and solution. So, what are the limitations of conventional dosage forms? All these four keywords which I have highlighted in yellow, they are not possible in case of traditional dosage forms. So these are the limitations in case of conventional or traditional or classical type of dosage forms. So few novel dosage forms available in the market are transdermal patches, implants, nasal system, microcapsules, microspheres, liposomes, phytosomes, and others. So very beautifully this infographic on the left side explains the need and importance of novel drug delivery system. This is the original copyrighted infographics of the University of Alabama at Birmingham. I am thankful to it for this image which I have taken from internet while googling for the keyword herbal or normal NDDS. So you see the first stage, tiny, hollow, this one. How NDDS is helping the patient. Tiny hollow microcapsule, each smaller than a red blood cell, even smaller than RBC. They are created layer by layer in the laboratory. Each microcapsule is loaded with the potent cancer drug doxorubicin. So doxorubicin is the name of medicament API which is anti-cancer in nature. It has been capsulated, that is the technique is microencapsulation. Now each microcapsule is loaded with the potent cancer drug, this one, right? Millions of these microcapsules could be injected into the bloodstream, but they will not start acting, they will not release the drug. Millions of these microcapsules could be injected into the bloodstream. The durable outer layer, which is not the drug, excipient, the durable outer layer would keep the cancer drug away from healthy tissues as the microcapsule travel to the tumor site. And once the microcapsule reaches the site of action, that is the cancer site, using an ultrasound machine which is outside the body, doctors could trigger the microcapsule to burst or to rupture to release the drug when they reach a solid tumor, releasing the cancer killing drug. So how beautifully through some technology directly the drug which is anti-cancer, that is doxorubicin, directly acting or releasing or attacking the cancer cells and not the healthy cells. These are the importance of or the features or the advantages of the NDDS. Now you see the image on the right side. So different types of NDDS are there like dendrimer, liposomes, nanogels, metallic inorganic and others. This is the example of transdermal page, which is again a type of NDDS. World famous company Cipla is producing nicotine transdermal page, and they are recommended for those who are addict of cigarette or tobacco chewing or any product containing nicotine for their habit forming effects. So in this, what they are claiming, within 24 hours you will get 21 milligram of nicotine. So for 24/7 it will be fixed on your skin and it will keep releasing nicotine in a gradual manner. So the person or the addict who want to give up the nicotine product, they can have it on their skin and they will not have the craving for smoking or other material having nicotine. So this is the benefit of Nicotex, that is the brand name owned by Cipla, and the drug is nicotine in the transdermal patch. And they are also giving the release profile in this second stage. So total 3 types are there: step 1 of 3, step 3 of 3. There will be 2 also. So in this they are saying 7 milligram of nicotine will be delivered over 24 hours. So when you start giving up the nicotine, you would need probably more amount of nicotine. So it is having 21 milligram in 24 hours. Gradually the dose will be reduced when you will be able to cope up, or when you will not be able to show withdrawal symptom. So another beautiful example of NDDS commercially available in market. Another product is the Exelon patch, a transdermal patch. So within 24 hours, it will release 4.6 milligram of the medicament. So what is the brand name? Exelon Page 5. Company name Novartis. And the drug is rivastigmine, a type of NDDS. 5 patches are there. It is used for the treatment of mild to moderate dementia of the Alzheimer type. Right? So this is the product for memory loss, to gain the memory. So rates of nausea, the side effects. So rates of nausea and vomiting are remarkably reduced with once daily rivastigmine page. So suppose one is consuming orally this medicine, so there will be more nausea and vomiting. But if she or he is applying a page, there will be reduced nausea and vomiting episodes because of the page, and because of page is there. So it can be applied at any time of the day with or without food. Another is fentanyl transdermal system. So rivastigmine was developed by Martha Winstock Rosin of the Department of Pharmacology at the Hebrew University of Jerusalem, Israel. And this product technology was sold to Novartis by Yushum for commercial development. It is a semi-synthetic derivative of phytostigmine. So phytostigmine is an alkaloid obtained from plants, and its synthetic sibling is rivastigmine. It has been available in capsule and liquid formulation since 1997. But to minimize the dose and to minimize the side effect, transdermal page concept has been given. In 2006, it became the first product approved globally for the treatment of mild to moderate dementia associated with Parkinson's disease. And in 2007, the rivastigmine transdermal page became the first page treatment for dementia. So advantages of novel dosage forms already we have seen. We will now see the benefits from viewpoint of herbal, because we have to study herbal NDDS. But directly we cannot jump onto the topic. We should know some background. So I am giving you lot many examples and basics. So advantages are solubility and bioavailability of phytoconstituents is enhanced. Distribution and pharmacological activity is enhanced. Drugs can be released at the predetermined rate and deliver the drug at the site of action. Produces less toxicity as novel dosage forms avoid accumulation of drug to non-target areas. We have already seen directly drug will reach to the site of action in a in a I mean, you can even control the rate release and type of release profile. So there is no question of drug accumulation or drug fluctuation. Stability of phytoconstituents can be improved, easily administered to pediatric and geriatric patients. Phytoconstituents are susceptible to acidic environment and undergo liver metabolism. This is not the case in case of NDDS. So limitations of herbal dosage forms. Conventional dosage forms cannot exert effects for longer period of time. Means you cannot control the release profile. Once tablet is consumed, it will release as per the profile you are not having control on. Phytoconstituents cannot be released at desired site of action. Desired therapeutic response cannot be achieved. And finally, phytoconstituents have limited solubility and bioavailability when administered as conventional dosage forms. So in your syllabus, there are mixtures, herbal syrup, herbal tablets. So very quickly, we will talk about what is herbal mixture. Herbal mixtures are solid, dry powder consisting of more than 2 or 3 individual powders of the different plant material. and they are mixed, and before finally packaging they are properly mixed, triturated, and it is passed through sieve number 60 before packaging or before evaluation. So we have different types of powder in the market. For example, Trifla churna, amla powder, arjuna powder, licorice powder, sitapaladi churna means churna powder, or you can call it mixture. Mixture is allopathic term, English word, and churna is Hindi or Ayurvedic terminology for a mixture formulation. So there is no rocket science in formulation of mixture, and for evaluation of mixture, you have to go for various quality control parameters like morphological, that is the what we call it visual inspection, color, odor and taste. Then we can have the feel by taking between the finger and the thumbs, I mean first finger and thumb. Then in case then we can go for solubility, identification, taste, that is phytochemical screening, TLC, HPTLC profile, it is powder. So we can also go for different QC parameter of herbals which are exclusively designed or recommended for powder or solid, like ash value, which are of three type, that is total ash value, acid insoluble ash, and water soluble ash. We can go for extractive value, that is water extractive value or alcohol extractive value, in which we try to find the quantity of the powder which is soluble in the solvent, mainly water and alcohol. We can go for loss on drying, that is LOD, and we can also go for moisture content. So these are, and we can also go for angle of repose. So these are few recommended quality control or evaluation or assessment or standardization parameter of herbal mixture. Now we all know syrups are sugar solutions prepared either as per IP or USP. They are very thick, viscous solutions and syrupy in nature, and they are basically recommended for the geriatrics and pediatric patient. Geriatric means old age patient, pediatric means young patients, tiny toddler or children who are not comfortable in swallowing the tablet. So basically herbal syrups are basically recommended for treatment of cough. So you will have different types of herbal raw materials. You will either make the powder, or you will have the decoction. You will remove the solvent, then you will add this mixture into the syrup base, and you will deliver it to the patient. I mean, it is ready to be used by the patients after going through QC parameter. So what are the QC parameter? That is, QC means quality control parameter for syrup. They are basically viscosity, color, odor and taste, and stability profile. So these are common QC parameter for syrups, right? So syrup is a concentrated mixture of sugar in purified water. The oral use of liquid pharmaceutical has generally been justified on the basis of ease of administration. As I told you, they are easy to take for patients like old age and children who are not comfortable in taking the tablet type of or capsule type of dosage forms. Now basically the common ingredients in herbal preparation in herbal syrups are tulsi, licorice, ginger, black pepper, vasaka, and they are recommended for the treatment of cough. Now, right now, how to make syrup already we discussed. And herbal syrup is prepared by combining a concentrated decoction with either honey or sugar and sometimes alcohol. The base of such a syrup is a strong herbal decoction. Mixing a decoction with honey or sugar help to thicken the and preserve the decoction. Herbal tablets, it is all, they are also called vati, gutika, ghanvati, and simply tablets. So for making tablet, you should have different types of raw material, that is your excipients, and your active ingredient, that is of herbal origin. And you will mix all the all the materials along with the excipients. You will make semi-solid consistency. Then you will make powder, free flowing powder. If you are using the punch machine, then all your ingredients should be mixed properly, they should be dried properly, and they should be free flowing in nature. By adding binder and diluents, you will punch the tablet. If you are going for ghanvati or vatika or gutika, then you have to make it semi-solid in nature, and by rolling the semi-solid lump or mass between your palms, you can roll it, you can roll it cylindrically, and you will break it to make small tiny round shape of atom which is called vati. Now for evaluation of tablet, we have different types of parameter which are also available for allopathic or conventional tablet, like hardness, friability, dissolution, disintegration, content uniformity, wet variation, physical appearance, and of course assay to determine the presence of active ingredient. Now we will talk about herbal NDDS. So there are liposomes, nanoparticles, microspheres, and phytosomes. So what are liposomes? These are prepared by incorporating the active ingredient inside the microscopic double layered membrane, which is of phospholipid, also known as phospholipid vesicle. Nanoparticle, these are colloidal particles of the submicron size which act as carriers for drug molecules. These are used to target various sites in the treatment of cancer, as we just discussed, disease of the reticuloendothelial system, and enzyme replacement therapy in liver. Now we will talk about microsphere. These are small solid particulate carriers containing the dispersed drug particles either in solution or crystalline form. Microsphere are used as carriers for drugs and therapeutic agents, especially in cancer treatment and hormone disorder. Now we are coming to phytosome, because in your syllabus they have only mentioned phytosome as herbal NDDS. So there is issue why there is need of phytosome, because most of the herbal materials which are flavonoids are polyphenolics in nature. And they are flavonoids, so they are water soluble, so they are not easily bioavailable in the body. So to minimize the dose and to increase the bioavailability of hydrophilic or those herbal material which are poorly soluble in oil, means they are hydrophilic. So to decrease the dose of hydrophilic phytoconstituents and to increase the bioavailability, there is need of phytosome. So between all the phytochemicals, only those having an active hydrogen atoms like polyphenols, they can be integrated into a phytosome structure. An active hydrogen atom can form a hydrogen bond between the herbal derivatives and the hydrophilic part of amphiphile molecule. So there is the need for hydrogen bond formation between the herbal hydrophilic material, listen carefully, hydrophilic herbal phytoconstituent, and the amphiphilic molecule, that is phospholipid. The phospholipid here is used to bind the hydrophilic phytoconstituents. So such type of phytoconstituents are flavonoids or polyphenols. So polyphenols are the major group of phytoconstituents extensively found in plant-based foods. Essentially, they are found in conjugated forms composed of sugar residues attached to hydroxyl groups. However, the sugar residues may directly attach to an aromatic carbon. So examples of polyphenols are flavonoids, and non-flavonoids, they are mainly water soluble. So these are the examples of polyphenols, and all these are the right candidate for phytosomes. If a phytoconstituent is purely oil soluble and not soluble in water, then there is no need to make the phytosomes. So examples of polyphenols are like phenolic acid, stilbenes, lignans, apigenins, flavonoids, anthocyanins, and others. So what is the meaning of phytosome? Phyto means plant and some means something like cell-like or round structure. Phytosomes are also called herbosomes. They are vesicular drug delivery system enhancing the absorption and bioavailability of low soluble drugs. Phytosomes are complex of phospholipids and natural active phytochemicals bound in their structure obtained by the reaction between phosphatidylcholine or any hydrophilic polar head group and plant extracts in an aprotic solvent. These formulations exhibit improved pharmacological and pharmacokinetic properties as compared to prevalent preparation. Right? A higher absorption rate leads to a lower doses of active constituents for exerting a biological effect. So Phytosomes are also called herbosomes. They are also called phytophospholipid complex. So phytosomes help to pass through the outer membrane of gastrointestinal cells and reach their functional site. Due to this property of phytosome, the absorption of these components is greatly increased and their bioavailability becomes much greater as compared to the original component without phytosome structure. Phytosomes have been found to significantly enhance bioavailability of many plant extracts like curcumin from Curcuma longa, ginkgo biloba phytoconstituents, silymarin, ginseng, and polyphenolic compounds found in olive oil, green tea, and grape seeds. So, phytoconstituents of ginkgo biloba, silymarin from Silenium marianum, ginseng, and all these components have been successfully tried in the form of phytosome to increase the bioavailability and to decrease the dose. Scientists have successfully formulated phytosome loaded with quercetin, which is a type of flavonoid, using phosphatidylcholine. So this is the basic material, that is phospholipid or lecithin. This is required to make phytosome. So using phospholipid or lecithin or phosphatidylcholine and cholesterol, scientists have successfully made the phytosomes of quercetin. And this phytosome of quercetin resulted in its better release and bioavailability, thus proving its potential to be used in formulation of fortified food products containing water insoluble antioxidants. So now we will talk about the chemistry of phytosome. How exactly hydrogen bonds are formed? So phytosomes, which are also called phytophospholipid complexes, are the vesicular system formed by the interaction between hydrophilic parts of the phospholipid. So phospholipid is a chemical moiety in which there are two features. The components have hydrophilic part also, which is called the head, and hydrophobic, that is lipophilic part, which is called the body or tail portion. So there is interaction between the hydrophilic head part of the phospholipid, which is called choline, and the hydrophilic phytoconstituents, and this results in the formation of hydrogen bond between them, and this is then surrounded by the lipophilic part of phospholipid, that is phosphatidyl. Right? This is how phytosomes are formed. The structural differences between liposomes and phytosome is that liposome have their active ingredient inside the hydrophilic cavity, but in case of or within the layer of membrane, while in case of phytosome, those components, that is the your excipient or the phospholipid components, are the part of membrane itself. Phytosomes are synthesized by complexing these active ingredients with phospholipids in the defined stoichiometric ratios under predefined condition. So if you want to know what is lecithin, you can read this one, and this is different types of definition. I mean, what do you mean by phospholipid, what is lecithin, what is phosphatidylcholine? So several methods are available for the synthesis or formulation of phytosome, such as the rotary evaporator method, anti-solvent precipitation method, freeze drying, co-solvency, and salting out technique. The main methodologies for the preparation of the phytosomes are shown here. So first of all, the popular and commonly used technique for producing phospholipid complexes are the evaporator approach and solvent evaporation. In the process of solvent evaporation, lipid material, that is lipid materials are dissolved in an organic solvent because lipids are soluble only in organic solvent like hexane. So lipid materials are dissolved in an organic solvent which was then removed by vacuum rotary evaporator. By the anti-solvent precipitation technique, preparation of Lawson loaded phytosome is done. Lawson is a active chemical constituent from the henna plant. So anti-solvent precipitation technique has been used successfully for preparation of Lawson phytosome. In this process, dichloromethane was refluxed with Lawson and soya lecithin. So lecithin is a type of phospholipid, and the major source of lecithin is soya, that is soybean. In this lecithin, addition of lawsone was done in the solvent, which is dichloromethane. Lawsone is a phytoconstituent which is soluble in water. Then to get the precipitate, which is stored overnight in vacuum, and N-hexane was added to make the precipitate. So usually in protic solvents like methanol and ethanol, at least one hydrogen atom is directly connected to an electronegative atom. Some liposomal drug complexes act in the existence of buffer solution or water, where the interaction of the phytosomes with the solvent occurs with a decreased dielectric constant. So what are the advantages of phytosome? Phytosomes can give protection against destruction of herb by digestive secretion. So basically hydrophilic drugs can be destroyed or easily metabolized by the digestive secretions, acids and gut bacteria. So to prevent this degradation, there is need for phytosome, and phytosomes are basically designed for hydrophilic phytoconstituents only. So proper drug delivery at site of action, the nutrient safety of herbal drug is retained, reduction in the dose, enhanced absorption and bioavailability of active principle, entrapment efficiency is high and predetermined, better stability due to formation of chemical bonds. Chemical bond between the hydrophilic part of the phytoconstituents, that is, I mean, your entire hydrophilic part and the hydrophilic part of the phospholipid, that is the choline portion, that is the head portion of the phospholipid. So phosphatidyl used in process act as a carrier, hepatoprotective and nourishes the skin also in some cases. So already we have seen the difference between liposome and phytosome. In case of phytosome, the hydrophilic material is part of the phospholipid. In case of liposome, your hydrophilic part is completely covered or encapsulated by the liposome material, that is phospholipid. This is how you can understand the process of liposome. So for example, I mean phospholipid. This is entire phospholipid and this is your hydrophilic plant material for which you have to make the phytosome. So this is the phosphate group, this is your hydrophilic part of the phospholipid, and this is non-polar or the hydrophobic or lipophilic part, right? So your this hydrogen bond of the hydrophilic material will react here and this will make one hydrogen bond, right? So this hydrophilic phytoconstituent become the part of entire structure, and in this way this is the rough structure, this is phospholipid and this is your drug molecule, that is phytoconstituents which is hydrophilic. So this becomes attached with the choline, this is choline part, that is hydrophilic, and this is the remaining part, that is phosphatidyl, right? Okay, so this will become the phytosome. Evaluation of phytosome. So different techniques are there for checking the efficiency of the phytosomes. So there are physical size, membrane permeability, percentage of entrapped solute and chemical composition of materials. The phytosomes are evaluated for vesicular size, zeta potential, entrapment efficiency, transition temperature and surface tension. So best example of commercially available phytosomes is silymarin phytosome, which is obtained from milk thistle, and this is the world famous drug for treatment of hepatic issues. So silymarin phytosome showed increased bioavailability due to enhanced lipophilicity and therapeutic effects than silibin alone. Silymarin phytosomes also exhibited better hepatoprotective activity than silymarin. As per one study, absorption of silibin from silibin phytosomes was found to be 7 times greater as compared to the absorption of silibin from regular milk thistle. Milk thistle is the name of plant in English which gives us silymarin. Another example is polyphenols of green tea. Another examples already we have discussed: quercetin phytosomes, grapeseed phytosome, then ginkgo phytosome, and phytosomes of curcumin and phytosomes of naringine. These are the comparative evaluation of phytosomes, liposomes, neosomes, ethosomes, and transferosomes in nano delivery system. This is just for your knowledge. Another way to understand how phytosomes are prepared. So analytical methods or quality control or evaluation parameter for the phytosome. So you can check their size and shape. You can go for determining surface charge, chemical composition, then lamellaricity and stability, encapsulation, and other parameters are there. So for size and shape, you can go for DLS, SEM, TEM, optical microscopy, and different techniques are there. For chemical composition, you can go for FTIR, NMR, mass, and different types of sophisticated analytical techniques. So these are commercially available phytosomes in the first column, available in market. In second column, name of company is there. In third column, what are the phytoconstituents for which phytosomes have been formulated. The second column is the name of company, and let me tell you, Indena is the first company in this world which has given the term phytosome, and lot many patents have been filed by this company, and most of the commercially hit, commercially successful phytosomes are given by Indena in this world, right? So these are different formulations, herbal NDDS, and these are commercially available phytosomes of curcumin, available on 1 mg. So with this, I finish this topic. Thank you very much.